if you see the discovery of different plant growth regulators, the plant growth regulators discovery is all the plant growth regulators are discovered accidentally. Here, first of all, auxins. Here, auxins were firstly observed by Charles Darwin, Francis Darwin. Charles Darwin and Francis Darwin, they observed the coleoptile of canary grass. So it is coleoptile. The coleoptile means in monocot plants, in monocot embryo, here plumule is here plumule radicle. The plumule is covered by a coleoptile. Radicle is covered by coleorhiza. It is coleoptile. It is coleorhiza. So the coleoptile in monocot plants only coleoptiles. Here coleoptile and coleorhiza are present. Here these scientists observed the coleoptile tip when it is exposed to unilateral light. When it is exposed to unilateral light, here it is showing the bending. It is bending towards the light. Next, here it is showing the response to light. So phototropic movement. It is a phototropic movement. Phototropism. So when it is when the coleoptile tip is removed, when the coleoptile tip is cut, it is the blunt end. So when the tip is removed, here it is exposed to unilateral light. Then here it is not showing the bending, no response to light. Next, the coleoptile tip is in another experiment, the coleoptile tip. Here it is covered with a cap. It is completely, the tip is covered with a cap. Then it is exposed to unilateral light. Only one side it is exposed to unilateral light. So then also it is not showing any response to light. So no curvature, no bending. No bending occurs, no response, no phototropic movement. So here later a number of experiments, they concluded that in the presence of light, the tip, the coleoptile tip, in the coleoptile tip something is produced. Here this tip is it is receiving the light, or it is showing the response to light. So in the presence of light only, here something is produced, and this chemical or this component, it is responsible for the here bending. So when it is when this tip is not present, here also same, but the tip is not there. When the tip is removed, here it is also exposed to unilateral light, but no bending occurred. So in the tip is receiving the light, and in the presence of light something is produced. That chemical or that component is responsible for the bending. So these scientists concluded that the coleoptile tip, here it is a canary grass coleoptile. Darwin scientists observed the canary grass coleoptile tip, and in the presence of light something is produced in the tip, and that chemical or that component is responsible for the bending, or that is responsible for the phototropic movement. Next, after some years, F.W. Wendt scientist, F.W. Wendt, Wendt scientists observed the oats coleoptile. Oats scientific name Avena sativus. It is also monocot plant. The monocot plant coleoptile, from this tip he extracted or isolated the auxin. So the first auxins were firstly isolated by Wendt from oats coleoptile. Next, gibberellins.